Today, we're diving into a high-stakes molecular detective story straight from the World Health Organization's 5th edition classification of tumors. We're looking at Philadelphia chromosome-positive ALL, or formally B lymphoblastic leukemia/lymphoma with BCR/ABL1 fusion. This isn't just a rundown of medical facts. It's a masterclass in how understanding cancer at the absolute microscopic genetic level has completely transformed the way we fight it. Let's quickly run through today's roadmap. 1. The Philadelphia chromosome story. 2. Anatomy of a genetic typo. 3. Diagnosing the disease. 4. Targeted therapy revolution. 5. The gatekeeper mutation. And finally, 6. The future of precision medicine. Section 1. The Philadelphia chromosome story. Let's rewind the clock back to 1960. Researchers in Pennsylvania made a truly groundbreaking discovery, the very first recurrent chromosomal abnormality ever found in human cancer. This led us straight to our main suspect. Philadelphia chromosome-positive ALL is a highly aggressive precursor B-cell leukemia. Its defining feature, a specific translocation, T922q34q11.2 between chromosomes 9 and 22. This swap produces a mutant fusion gene called BCR/ABL1. And unlike chronic myeloid leukemia, which originates in mature myeloid cells, this disease originates from lymphoid precursors. It's a dangerous genetic fusion, and it absolutely demands highly targeted therapy. When you break down the demographics, there's a pretty stark contrast. In kids, this specific fusion only drives about 3 to 5% of childhood B-ALL cases. But in adults, it's a massive 20 to 30%. And that frequency only goes up as patients get older, often bringing with it higher white blood cell counts and a much higher risk of central nervous system involvement. Section 2. Anatomy of a genetic typo. So how does this chromosomal culprit actually operate? Let's break down the mechanics. It's literally the weaponization of a gene. Normally, the ABL1 gene on chromosome 9 acts as a tightly controlled switch, regulating the cell cycle and apoptosis. But when it fuses with the BCR gene on chromosome 22, that switch gets broken. It gets stuck in the on position, what we call being constitutively active. It's constantly firing off oncogenic signals through pathways like RAS and PI3K, driving relentless, uncontrolled leukemic growth and refusing to let those rogue cells die. But here's the catch. Not all fusions are identical. The exact breakpoint in the gene matters immensely because it produces different proteins. The P190 transcript is the primary villain in ALL, showing up in 70 to 80% of cases, and it's highly lymphoid. The P210 transcript, on the other hand, is much more common in CML, though it can occasionally cross over into ALL territory. Section 3. Diagnosing the disease. Because this leukemia operates so stealthily, diagnosing it means we have to hunt for its specific fingerprints using highly advanced tools. Under a microscope, you honestly wouldn't know the difference. The blasts just look like conventional precursor B-ALL, small to medium cells where the nucleus takes up almost the whole cell. Morphology simply cannot diagnose PADS plus ALL. So we rely on flow cytometry to light up a specific precursor B-cell barcode. We're looking for heavy positivity for CD19, CD22, CD79A, CD10, and TDT. Now consider the sheer sensitivity required to track this disease today, 10 to the negative 6. That is literally 1 in a million. Using next-generation sequencing, we can monitor minimal residual disease, or MRD, at that astonishing depth. Finding even a single surviving leukemic cell among a million healthy ones is absolutely crucial because persistent MRD is one of the strongest predictors of a relapse. Before treating, we have to be certain of our target. It's critical to distinguish PH plus ALL from CML that has progressed into a lymphoid blast phase. Why? Because the treatment strategies are drastically different. In PH plus ALL, you usually won't see a prior history of CML, there's no preceding chronic phase, minimal basophilia, and, as we just mentioned, you're usually dealing with the P190 transcript. Section 4. Targeted therapy revolution. Once we've positively identified P plus ALL, it's time to deploy the countermeasures. And this is where the story gets truly incredible. Before the era of targeted therapies, the 5-year survival rate for adults with this aggressive disease was incredibly grim. It sat at less than 20%. Traditional chemotherapy simply wasn't enough to overpower that stuck-on switch driving the cancer. But science fought back. We entered the era of tyrosine kinase inhibitors, or TKIs. First came Imatinib, a total game changer and the first successful targeted therapy. Then the second generation, like dasatinib, noletinib, and bucitinib, brought even greater potency and better penetration into the central nervous system. And finally, the third generation arrived with ponatinib, designed for the absolute most stubborn, resistant forms of the disease. Section 5. The gatekeeper mutation. But biology is fiercely adaptable. Just as we gain the upper hand, the leukemia evolved to fight back. The stakes skyrocket when the cancer develops a resistance mechanism in the ABL kinase domain. The most infamous of these is known as the gatekeeper mutation, or T315I. This single microscopic mutation is a massive threat. It changes the shape of the cancer's protein just enough that our first and second generation weapons literally just bounce off. They can no longer bind and shut off the switch. This means doctors have to constantly monitor the patient, quickly identify this mutation if it appears, and immediately pivot to ponatinib, which actually retains the ability to bypass the gatekeeper. Section 6. Future of Precision Medicine. We are no longer just throwing generic toxins at the body and hoping for the best. A modern hematologist's clinical playbook requires immediate, simultaneous action. The second that fusion is diagnosed, you promptly start a TKI right alongside traditional chemotherapy. You immediately establish a baseline using quantitative RT-PCR, and then you continually monitor that MRD. If transcript levels start to rise, it's a huge red flag to check for mutations like T315I. The future here is incredibly bright. The current frontier lies in pairing these TKIs with brilliant immunotherapies. Take blinatumomab, a bispecific antibody that literally drags the body's own T cells right to the CD19 markers on the leukemia cells. We're also seeing incredible results with inotuzumab ozogamicin and revolutionary CD19 CAR T cell therapy. Combined with a TKI, patients are achieving excellent MRD clearance. In many cases, it's improving survival so much that some patients might even safely avoid the grueling process of a stem cell transplant entirely. This expert perspective directly from the WHO 5th edition perfectly summarizes our journey today. Modern management integrates genetics, MRD monitoring, tyrosine kinase inhibition, and immunotherapy. It represents the absolute pinnacle of precision medicine in hematology, showing exactly how far we've come from a generic diagnosis to a highly individualized cure. And that leaves us with a thrilling thought about the future of oncology. If we can map out and neutralize a genetic typo this complex, what other doors is precision medicine about to unlock? How close are we to permanently shutting off the genetic switches of all hematologic malignancies?